Hey, welcome to all of you to my channel, that's Meets Bio World. So today we are going to study about glycolysis, one of the most important topic from the unit of bioenergetics and metabolism. Now, what is glycolysis? The topic name is glycolysis. Now, firstly, we should try to understand what glycolysis means. Now, this glycolysis, it simply it refers to the breakdown of a sweet substance, because in the Greek language this glycolysis term has been derived from the two words. So this glycolysis from the Greek, it has been derived from the two words. One is glycus and another is lysis. So this glycolysis is in the Greek language, it has been derived from the two words known as glycus and lysis. Now, what glycus means? Glycus means the sweet substance, and lysis means the breakdown or splitting. So the glycolysis simply means the splitting of a sweet substance. Now, here the sweet substance is referring actually to the sweet substance is referring towards glucose. So here the glucose is actually the sweet substance which is getting splitted through an enzyme catalyzed, through a number of enzyme catalyzed reactions are occurring in this glycolysis. And in this glycolysis, the sweet substance which is known as the glucose is getting splitted up through enzyme catalyzed reactions. Okay, now this glycolysis, if we move on further, this glycolysis is also known as the Embedden-Meyerhof pathway. So the another name for the glycolysis is Embedden-Meyerhof pathway. So Embedden-Meyerhof pathway is another name of glycolysis. Okay, so why this Embedden-Meyerhof pathway is known as glycolysis? Why glycolysis is known as Embedden-Meyerhof pathway? Because it was the Embedden and Meyerhof, these are the two prominent scientists who discovered this process of glycolysis. Okay, so that's why it is also known as Embedden-Meyerhof pathway. Actually, these are the two scientists which have just discovered this process of glycolysis. Now, what is happening inside the glycolysis and what process is like it? It is actually an oxidative process. So glycolysis is an oxidative process. So what oxidative process means? Now, oxidative process means in which there is addition, or in which simply we call that as in which there is breakdown of any substance, with the involvement of oxygen. That's called oxidative process. So oxidative process means it is simply a splitting process in which the entry of the oxygen will lead to the breakdown of the bonds there. Okay, so oxidative process, and in this oxidative process what is happening up? One glucose molecule is getting partially oxidized into 2 moles of pyruvate. So what is happening? It is an oxidative process in which 1 mole of glucose is getting partially oxidized. That means it is not completely getting completely this split. It is not glucose is not completely getting splitted up. It is partially getting splitted into 2 molecules of pyruvate. Okay, 2 molecules of pyruvate. So this is known as the oxidative process. So glycolysis is an oxidative process in which 1 mole of the glucose is getting converted into the 2 moles of the pyruvate through a series of the enzyme catalyzed reactions. So this process is not a single step process, it is a multi-step process. It is a 10-step reaction, and in this 10-step reaction the 1 molecule of the glucose is getting ultimately finally getting converted into the 2 moles of the pyruvate. Okay, now moving further. Now why this glycolysis occurs, this is the another thing, this is a very important thing. So we should always know that occurrence, why this glycolysis, why this process is occurring. It is occurring inside the cytoplasm or the cytosol of the cells, all cells. So why this glycolysis is occurring? It is occurring inside cytosol. So what is cytosol? Cytosol, inside the cell, inside the plasma membrane, or simply inside the cell, there is a fluid like material known as the cytoplasm. In that cytoplasm there are organelles there, but apart from the organelles, the fluid part is known as the cytosol. So inside cytosol of all cells. So this glycolysis is occurring inside the cytosol of all cells. And why there is all cells? Why it is written all cells? Why not cells? All cells means prokaryotic as well as eukaryotic. Okay, good. Now it is one of the unique pathway. Glycolysis, it is one of the unique pathway which occurs in both aerobic as well as the anaerobic conditions, and it does not involve the molecular oxygen. Okay, it does not involve the molecular oxygen. It is not dependent on the molecular oxygen. It's a unique pathway. It will occur in presence of oxygen as well as in absence of oxygen. Okay, so unique pathway. This is a unique pathway. Now what is actually happening in the glycolysis? How these I have just mentioned about that. It is a 10-step reaction. It is basically a 10-step reaction in which the 1 glucose molecule will get converted into 2 molecules of the pyruvate. So but this is not a single step reaction, it is a 10-step reaction. So if we go to the mechanism of glycolysis, now the mechanism of glycolysis is simply, firstly we should note that it is a 10-step reaction. 10-step enzyme catalyzed reaction is a series of enzyme catalyzed reactions. So 10-step reaction. Now I am reactions I have abbreviated this as RXN. Okay, it's a reaction. Now the first step is, the first reaction or the first step of glycolysis is phosphorylation. Now who is going phosphorylated? That means the first step starts from the glucose, because the substrate which is going to convert into the pyruvate is glucose. So the glucose, it will get converted ultimately finally through 10-step reactions, through 10 steps, lastly finally into the pyruvate. But at every step, how this process is going on. Now the first reaction is the glucose, 1 molecule of the glucose is getting converted, into glucose 6-phosphate. Glucose 6-phosphate. Okay, now this reaction is actually called as phosphorylation. This reaction is actually called as phosphorylation. Now this first step is known as phosphorylation. What is happening in the phosphorylation? Actually the glucose is phosphorylated by ATP. So there is involvement, there is investment of energy, there is use of energy in this step. The step is utilizing energy. So ATP is being utilized for what purpose? when ATP gets utilized, when ATP breaks down, it gets converted into ADP plus inorganic phosphate. Okay, and this use of the energy is to simply add the phosphate group to the glucose, so that glucose will get converted into glucose 6 phosphate. So this phosphorylation is simply utilizing of ATP to form the glucose. Basically the phosphorylation, we can simply, what is phosphorylation here? It is simply adding up the phosphate group towards the glucose. But how the phosphate will get added to the glucose? It is basically, there is involvement of the energy ATP, and ATP on breakdown it will get converted into ADP plus inorganic phosphate, and one of the phosphate will get attached towards the glucose. Now this negative charge of the glucose, this negative charge of the phosphate, not the glucose, this negative charge of the phosphate, it will prevent the passage of the glucose 6 phosphate through the plasma membrane. So negative charge, what is happening in the negative charge of the phosphate, it will prevent the passage of glucose 6 phosphate through the plasma membrane. So when the passage means that the glucose will not go outside the membrane, it will not go outside the cell. Why? Because who is preventing the glucose from going outside the cell or while crossing the membrane? It is basically the phosphate. The phosphate which gets attached towards the glucose through the phosphorylation reaction. Now this phosphorylation reaction, this adding of the phosphate with the glucose, it will prevent the glucose in going out of the cell. Okay, because of the negative charge of the phosphate. So due to the negative charge of the phosphate, it will prevent the glucose in going out of the cell through the plasma membrane. So that means the glucose gets trapped, it gets trapped inside the cell, it gets trapped inside the cell. Okay, now this reaction or this step in which the glucose is getting phosphorylated and glucose is getting converted into the glucose 6 phosphate. Now this reaction is irreversible reaction. And one more thing I will add here, that every reaction, it's a metabolic reaction, and we know that metabolic reactions are simply those reactions which are happening inside the body. Okay, now every reaction is catalyzed by an enzyme. So this reaction is also catalyzed by enzyme. And which enzyme is catalyzing this reaction? The enzyme which is catalyzing this reaction is known as hexokinase. Okay, hexokinase. So hexokinase is catalyzing this reaction, and this is an irreversible step. So this reaction is irreversible step. So is an irreversible, okay, irreversible reaction. What irreversible reaction means? Irreversible reaction means that from the glucose 6 phosphate has been formed, but glucose 6 phosphate will never get converted back into the glucose. That's called as irreversible reaction. Okay, that is why it's called as irreversible reaction. Okay, now this was the first reaction in this glycolysis. The first reaction is glucose getting splitted up or glucose getting converted into glucose 6 phosphate through a reaction known as phosphorylation, and this reaction is catalyzed by hexokinase enzyme. Okay, and there is involvement of ATP. And what is phosphorylation and why there is involvement of ATP? Phosphorylation, in the phosphorylation reaction, simply the glucose is getting attached, the phosphate has been attached towards the glucose, and this wire from this phosphate came up. This phosphate came from the ATP. Okay, so this was the first reaction in this process, that is conversion of glucose into glucose 6 phosphate. Now the second reaction is when the glucose is getting converted into fructose 6 phosphate. So glucose, the second reaction, glucose 6 phosphate, the glucose 6 phosphate here, the glucose 6 phosphate is getting converted. I can just draw it here. the glucose 6 phosphate is getting converted into fructose 6 phosphate. Now what is happening here? Only the glucose is getting converted glucose 6 phosphate. That means glucose here is getting converted into fructose. So it's a kind of an isomerization reaction. And the second reaction is known as isomerization reaction. So now the second reaction is isomerization reaction. So in this isomerization reaction, what is happening up? The glucose 6 phosphate is getting converted into fructose 6 phosphate. Okay. Now how is basically there is reversible rearrangement of chemical structure. What is isomerization? What is isomerization? In order to make your concept much stronger, you have to just understand that every point. You have to understand every point. And here I have just noted isomerization. Now what is isomerization? It is simply the reversible rearrangement of chemical structure. It is reversible rearrangement of chemical structure. Okay, reversible rearrangement of chemical structure. So that means this reaction should be reversible, because glucose 6 phosphate, it gets reversible rearrangement reaction, that is isomerization, and gets converted into fructose 6 phosphate. But it's a reversible reaction. Isomerization is a reversible reaction, and that means if there will occur the rearrangement back, then the fructose 6 phosphate will get back converted into the glucose 6 phosphate. Okay. Now now the enzyme which is catalyzing this reaction is known as phosphoglucoisomerase. So which enzyme is catalyzing this reaction? I am writing here phosphoglucoisomerase. Okay, so the second step gets completed up here. Okay, so for the today we will study for today up to this step, and the next 8 reactions we will study it tomorrow. Okay, so just focus on these things, just revise these things up, and it will surely will be asked in the examination, and it will surely help you.